So it's my pleasure to introduce Vector Space Biosciences to give a chat to the AWG about their computational approaches and their thoughts about. Really look forward to hearing what they have to say today. So with that, I'll turn it over to you guys.
Hi, this is Kajan.
Kajan, hi.
Hi.
Welcome.
Well, let me go ahead and share our screen here. And we'll start with the title, Using Scientific Data Engineering Pipelines to Accelerate Novel Discoveries in Space Biosciences. And I'd first like to introduce our team. This is a team that has spent time at Genentech. That includes myself and a few other members, up to Lawrence Berkeley National Lab, the Biosciences division there. And I'll just take you through a quick history. We got our start in the 1990s at Genentech working on HER2. This was related to breast cancer research and language processors associated to building what are called silk file formats. This related to pharmaceutical recipe builders that Genentech used back then. We spent a few years in Silicon Valley, and that was about the time that XMine, a small bioinformatics company, hired us to engage in language modeling. This was related to bioinformatics and early NLP, latent semantic indexing, inductive logic reasoning. This was all based on vector representations or embeddings. And the result of this, I'm just going to run through this real quick, was a patent related to inter-term relevance for analysis for large libraries, also related to language modeling. And there was a product that was produced called Opus. Opus was a way for scientists to relate genes, drugs, diseases with one another. And this was eventually purchased by NEC in Japan. They wrote a paper titled BioCompass. You can look that up when you want. We'll hand the presentation out at the end if anybody would like to see it. We were then invited to Lawrence Berkeley National Lab. This was within the Bissell Lab to establish new ways of developing relationship networks between proteins, drug compounds, and this was connected to the following report produced by Mina Bissell, commissioned by NASA Ames Research, titled Modeling Human Risk, Cell and Molecular Biology in Context, a report on animal experimentations at the frontiers of molecular and cellular and tissue radiobiology. You can read more about it again in our presentation here. This involved several national laboratories, and part of our work also included breast cancer research based on the extracellular matrix and the tumor microenvironment and radiation damage to chromosomes. We also did research in genes, which extended the lifespan of nematodes. We also used a variant of AutoClass. This was the first artificial intelligence software to make a published astronomical discovery, part of NASA's first AI program. We used it to compare supervised vectors with one another, and we converted it to an unsupervised variant, which used continuous values. This was also related to latent semantic indexing and early natural language processing and natural language understanding. Some of our work included results interpretation related to directed acyclic graphs, or DAGs, and knowledge graphs, along with their visualization and interpretation. We also created relationship networks using vector representations or embeddings, and that's based on early language modeling. This led to a patent related to system and method for generating a relationship network out of Berkeley Lab, including products for industry related to search engines. This also resulted in a paper related to extending the lifespan of nematodes based on analysis of their genes within literature. Eventually, Berkeley Lab took a 5% stake in our spinoff, SeqPod. It was, quote, titled One of Berkeley Lab's Success Stories. And that eventually led to consulting with the US Navy's Space and Naval Warfare Division related to knowledge discovery dissemination. And that involved a few government agencies, also in our presentation that you can review at any point in time. Again, based on relationship networks, networks based on hidden relationships between entities. Eventually, we left the lab to start a company in industry. The company's name was SeekPod. We powered Spotify back then. And in 2013, Word2Vec was published. This was a language model or based on vector representations or embeddings. And of course, that led to the seminal paper, Attention is All You Need, in 2017, based on the transformer model. And today, we operate with language models, advanced forms of language models, which include natural language processing and natural language understanding. So that gives you a brief history of who we are and why we're here. We also maintain a network of funders in the financial markets, along with a network of scientific collaborators and labs with a multi-omic biological focus on stressors related to the tumor microenvironment, the brain extracellular matrix, exosomes, mitochondrial stress, muscle atrophy, and human aging. Our goal is to investigate countermeasures associated to diseases related to stressors during spaceflight, which can double as therapies resulting in precision medicine for all humankind. Many might ask, well, how do you do that with language models alone? Well, by using scientific data engineering pipelines coupled with language models, which are trained to detect hidden relationships between genes, proteins, and pathways, including compounds, molecular sequences, and signatures. This also includes deep knowledge domain expertise, human-in-the-loop curation, and experimental validation, of course. So as many know, today's artificial intelligence and machine learning operations accelerate research. Language models, however, sit at the tip of the spear. They represent the tip of the spear in today's AI and ML operations, and we'll explain how. Language models, of course, can be used to analyze human language or ancient human language. In the case of epigraphy, for example, understanding inscriptions or deciphering inscriptions. Modern human language might include dictionaries, encyclopedias, ontologies, annotations, long and short descriptions based on data sources from clinicaltrials.gov, Drug Bank, or PubMed, as we're all familiar with. Now, what most don't really know is that language models can be applied to non-human language. And going back in time, when you look at non-human language, we were able to apply language models to sequences of objects. This was at Berkeley Lab in 2005. And when you look at language models produced by Thomas Mikolov while at Google related to word representations in vector space, language models can be used beyond words, as that paper also noted. And of course, Attention is All You Need, the seminal paper by Vashwani, brought out and proved that language models can be used for all kinds of data. The illustrated transformer or transformers from scratch, word embeddings, deep learning to annotate the protein universe. When you look at DNA sequences, for example, look at number 7 here, this reference, DNA sequences perform as a natural language processing by exploiting deep learning algorithms to identify methacetylene. And when you look at de novo molecular design and chemical language models, when you look at sequence-based protein structure prediction using language models, you then eventually wind up at AlphaFold 2. At the heart of AlphaFold 2's breakthrough in predicting the way protein folds, based on a sequence of amino acids, at the core of that is the language model, or the transformer, or multi-head attention encoder decoder stack, all powered by vector representations or embeddings. This is an article produced by Oxford Protein Informatics Group titled AlphaFold 2 is Here, What's Behind the Structure Prediction Miracle? And as you can see with this graphic here, you can see that vector representations or embeddings are used to predict similarity between protein structure or protein sequences, also based on vector representations, the transformer, and embedding, and in fact, natural language processing beyond words. When you look at transformer models today, many of you that are familiar with AI and ML, and most people on the technical side that have heard about advances in artificial intelligence have heard about BERT or GPT-3 or DALL-E, being able to use text to generate images, for example. finding patterns in images. To the right, you see a group of language models. These language models represent some of the latest, and this list continues to grow. Again, showing that language models are the tip of the spear in AI and ML operations. So how can scientific data engineering pipelines and language models actually accelerate the generation of new hypotheses, new insights and interpretations, or new discoveries? Well, we have a couple of ways. The first would be to consider that every 24 hours, about 4,000 new papers are submitted to the National Library of Medicine's PubMed. And out of those, about 1,500 are new submissions. The remaining are modifications or retractions. And the results include real-time correlation matrix data sets. And if we can show you what our real-time log looks like here, we bring in every 24 hours about 1,500 new PubMed IDs. And you can see the process happening in real time right here. When that happens, we produce input files and then correlation matrix data sets. So you can see that if a paper comes in talking about, or if a paper is published that's submitted to PubMed, mentioning a relationship between a protein and a drug compound, a new relationship, you might want to capture those because there are a lot of papers being published every 24 hours. And that includes papers related to drug compounds, diseases, and other related biochemicals and sequences. So we try to capture those. And the only way to do that is with a language model and in real time. So what are other ways that scientific data engineering pipelines and language models can accelerate the generation of hypotheses, new insights and interpretations or discoveries? Well, when you look at genes, proteins, and pathways, they can be represented by feature vectors or embeddings. And that results in the ability to engage in similarity measurements between proteins, compounds, and sequences. That, in turn, provides the ability to detect hidden relationships between proteins, compounds, and sequences, and then the extraction of those relationships before they are published. And that's key, because what you can do is, as shown in this graph here, you can show that there are relationships between a protein and another protein, or between a protein and a drug compound, that can be detected based on language modeling by measuring feature vectors or the embeddings for similarity. In this case, we're measuring similarity between two AWG. These are muscle atrophy genes associated to the stressor microgravity, PARP1 and INFAT5. The relationship converges over time from 2010 to 2015, as you can see here. And that's based on analyzing the literature in real time. This enables the prediction of a future or latent relationship between proteins until that relationship is made public or published. And you can see, looking to the right here, that the relationship was first published in 2017. So from 2010 to 2015, we're showing that the relationship grew stronger over time and eventually converged to an explicit published relationship. And we have another example here of TF and INFAT5, measuring similarity between them, converging over the years 2010 to 2015, and finally with a published paper showing an explicit relationship in 2018. So again, a future prediction between two proteins. So what are other ways that scientific data engineering pipelines and language models can accelerate the generation of new hypotheses and insights, interpretations, and discoveries? Well, when you look at the way that you can structure relationships between proteins, compounds, and sequences that are in a context-dependent way, you can build networks. You can construct networks beyond knowledge graphs, where the networks contain hidden relationships between proteins and compounds and other proteins and diseases and so forth. These networks can change over time based on user-defined context. And of course, as mentioned, they can also be real-time. And we'll show you a quick demo. We've built a product around this, and this product relates to a protein network. This is just an animation that you see here. We'll take you to the real product here, and you can see we've queried for INFAT5, the AWG gene, and INFAT5 has a number of proteins related to it within this network with scores associated to it that show the strength of relationship. And as you can see at the top here, there are known connections and there are hidden connections. However, the real power of this happens when you're able to construct a network of proteins related to NFAT5 in the context of heart muscle here. So having a context-dependent or user-defined context enables you to construct a network that's different from an ordinary network. So as you can see, the network for NFAT5, the general network for NFAT5 and its related proteins is much different than the network within the context of heart muscle. And of course, we have a few graphics here within our presentation that you can take a look at some point. One of the graphics we have relates to the sunburst image. And this sunburst, each sunburst is attached to a node so that you can drill down and determine how strong one node or one protein is related to another protein or compound. And of course, validate with external citations or references. So what are other ways that scientific data engineering pipelines can assist in generating new hypotheses, interpretations, or discoveries? Well, that would include by accelerating the process of discovering a drug that can be repurposed, or a therapeutic target, or a nutrigenomic cocktail, the development of a nutrigenomic cocktail, which can translate to countermeasures for diseases-associated distressors. One case study we have relates to Fanconi's anemia, and this relates to a language model being applied to biomedical data, resulting in a form of precision medicine. And of course, our goal is to double countermeasures as precision medicine for the benefit of all humankind. And in conclusion, our group, including its financial market partners, continue to fund operations in space biosciences. For more information, please contact us at support@vectorspacebio.science or myself at vectorspacebio.science or through our site, vectorspacebio.science. We have supplemental information associated to this presentation that includes details related to the actual input and output of a scientific data engineering pipeline, including the case study of Fanconi's anemia. So if you'd like to get a hold of this presentation and understand how language models are able to predict or assemble a sequence that might be valuable in terms of producing precision medicine, feel free to request the presentation at any point in time. We've got a detailed explanation related to how input files are created, not just for human language, but also for sequences, as you can see here with a DNA sequence related to NFAT5. And then, of course, we've got feature vectors. These feature vectors are the result of the data engineering pipeline, where these are actual embeddings you can take a look at, including embeddings for non-human language. So if you have any questions, feel free to contact us at any point in time or request the slides at any point in time. Thanks for joining.